Rice is an important food source for humans. However, it is susceptible to threats including pests, diseases, and environmental stresses. To overcome this problem, humans have been using traditional selective breeding method to cultivate the paddy species that has the most resilience to the threats. This method is effective, but it requires a lot of time and resources to achieve the goal. Luckily, in recent years, bioengineers have been able to create a genetically modified paddy plants through the integration of biotechnological advancements into rice cultivations. Genetically modified paddy plants are resistant to pests and diseases, have high tolerance to environmental stresses, and able to produce rice with higher nutritional value. Moreover, the efficient use of resources and reduced chemical inputs make genetically modified paddy plants a sustainable choice for modern agriculture.
There are three objectives for this discussion. The first one is to explain how gene technology can be applied in agriculture of paddy. The second is to evaluate the yield performance of GM paddy plants with desired traits versus conventional varieties under similar conditions. And the third is to identify the advantages and disadvantages of GM paddy plants.
Microprojectile bombardment is a deliver genetic material directly into target cell by bombarding them with microcarrier particle at high velocity where generated by either an electric discharge or helium pulse. The process involves preparing microcarrier particle typically made of gold or tungsten, coating them with desired genetic material and loading them into a gene gun. The gene gun shoots out code particle using pressure helium with a burst disc releasing gas to propel the microcarrier towards target cells. Target cell placed under vacuum are typically on a petri dish or culture plate. The high velocity particle penetrates cell membrane, delivering genetic material into the cytoplasm. Once inside the cell, the genetic material separates from the microcarrier, becoming available for cellular machinery leading to gene expression. GMO crops have been increasing yield for 20 years with more progress ahead. There are many ways to improve yield production nowadays that is resistant to insect and tolerant to herbicides. Based on our graph, we can see that the improvement of yield production in the latest few years, where the non-modified yield have a low percentage yield production compared to the modified yield. The difference production of yield between non-modified and modified yield may be because of several factors. As a result, it showed that the modified yield have a high production of yield that can reduce the rate of yield production in countries that have a food crisis, such as Central African Republic.
From the result, the reproduction of yield from paddy that are genetically modified is higher than normal transplant paddy. Genetically modified paddy plants are engineered to produce toxins that are harmful to certain pests, such as insects that feed on the rice plants. These developments have led to improved yield, quality, and tolerance to biotic and abiotic stresses. Using plant transgenic technology, researchers have transferred the Bt gene into crops to control target pests. So far, it has been reported that transgenic rice with the Bt gene can effectively control pests such as yellow stem borers, strep stem borers, and rice leaf rollers.
Genetically modified rice crop has been developed to address various challenges in rice cultivation, including pest resistance, herbicide tolerance, disease resistance, and improved nutrition. This development has led to increased yield, quality, and tolerance to biotic and abiotic stress. However, collaboration between scientists, policymakers, farmers, and consumers is essential to harness the full potential of GM technology in ensuring sustainable food production and agriculture development.